Good afternoon, everyone. My name is Jayla Burton, and today I'll be presenting my environmental chemistry research, our PFAS detector in shearwater birds. So, oops. So as Ellie stated, I am a rising sophomore animal science major at the illustrious Tuskegee University. I entered at the Lohmann Laboratory at the University of Rhode Island for my Ocean Explorers program, and I was mentored by Dr. Reiner Lohmann, and I worked directly with Taylor Elpers, Dr. Lohmann's graduate student. So, what have I been researching? During the past 9 weeks, I have been studying PFAS contamination in great shearwater birds, specifically by analyzing the feathers on different parts of their body to see how pollution accumulates. So PFAS stands for per and polyfluoroalkyl substances. And over here, I have an example of what PFAS looks like. So here are the most common PFAS. We have PFOS and PFOA. PFOS is has a sulfuric acid base, and PFOA has a carboxyl acid base. And what really makes these two compounds unique is their structure. So that long carbon chain surrounded by the fluorine base is really what makes these compounds very strong and difficult to break down in the environment, which creates a risk for both human and animal health. So many of the products that you use on a day-to-day basis contain PFAS. An example here shown is firefighting foam, as well as many other products. Do you guys use non-stick pans? Yeah, so those products contain PFAS inside of them, as well as other packaging material and waterproof gear. So PFAS could enter the environment in many different ways. One of the most common ways that PFAS could enter the environment is through firefighting foam. Once used, the firefighting foam sinks into the underground and contaminates the soil and underground water. From there, it is transported into the ocean and rivers, and it contaminates the living organisms within the ocean. The fish consume those living organisms, and birds, such as great shearwater birds, consume those fish. So even though great shearwater birds were nowhere near where the area where the firefighting foam was used, they still have PFAS inside of their bodies, which shows how mobile and persistent these chemicals are. So normally, scientists detect PFAS through using blood samples and animal livers in the birds, but my method is using feathers, which is non-invasive and it doesn't require the euthanization of the animals. And so here's a picture showing that. And now let's get into my feather extraction. So again, I conducted research on great shearwater birds. The birds migrate between the South Atlantic and the North Atlantic. They're like ocean nomads, so they spend a lot of their time in the sea, and they die after becoming entangled in fishermen's nets. So they're very dumb birds. And I worked in an EPA laboratory to remove the feathers from multiple body regions of the birds, and I removed the wing primary feathers. Here I show an example demonstrating that. So I removed the P3, P4, P5, and P8 of the bird's feathers, as well as the chest, tail, and back. So here are my quality control samples. So in each batch, I included a process blank and a process spike. A process blank is basically a sample that contains no analyte and it's run through the same exact process you would a feather sample. And it just checks for any contamination during processing. A process spike is a sample that has been intentionally spiked with a known amount of analyte, and that checks for any loss of analytes during handling. So here are my methods. So the first step that I did is I rinsed the feathers. After that, I sonicated the feathers. That's where I filled each feather up till 12 milliliters. And it's just basically a bath that just mixes the fluids together. Then after that, I let the samples air dry for 24 hours. And then after they're fully dried, I cut them into 1 to 2 millimeter pieces. So very, very tiny pieces because the surface area has to be very low to be able to extract the PFAS. Then after that, I weighed it until 250 milligrams per sample. So now let's get into my PFAS extraction. So I added methanol into all of my samples, and I placed the samples on a shaker table for 24 hours to just let the methanol absorb in the feathers. And then I centrifuged and collected the liquid supernatant. So a centrifuge is basically this really cool machine that basically allows my feather samples to sink into the bottom so that it's easier to conduct my methanol extraction. And so I repeated this twice more until I had 12 milliliters for each sample. So here's my PFAS analysis. So I spiked each of my samples with their internal standards, and then I diluted for the LCMS-MS analysis. The LCMS-MS stands for Liquid Chromatograph Tandem Mass Spectrometer, and that basically just measures how much PFAS I have in my samples. And then I integrated my samples using SciAx software. The software is supposed to integrate it by itself, but you have to manually do it and it takes hours. And so what are we measuring? So again, we have our carboxylic acids and then our sulfonic acids. You could see the carbon chain surrounded by the fluorine atoms, which makes the bond very hard to break down. And here are some of the compounds that you see in my samples, and they're very common to see in marine ecosystems. So here are my quality control results. So on the x-axis, you see, so I separated them from bird A and bird B. And on the x-axis, you could see the process spikes and the process blanks for both birds A and B. And then on the y-axis, you could see the concentration of nanograms per gram. And all the colors that you see are different types of PFAS. And so for our process spikes, I spiked all of my samples with 4 nanograms per gram, and you could see that I still have approximately that same amount, which means that my recovery is really great. And from my process blanks, you see no contamination, which means my samples aren't contaminated. And so here are my preliminary results. So on the x-axis, you could see all of my samples for both birds A and birds B. And then on the y-axis, you could see the concentration. Um, there's a lot of PFAS in my samples for my preliminary data. And so if you look at P3 for both birds A and birds B, you could see that it's kind of following a trend for each types of feathers. And then I separated PFOS because I wanted you guys to be able to clearly see the other compounds. And so here are my actual results. So we've lost a lot of the PFAS compounds after applying our quality control checks. And now we have PFSA, PF, FOSA, and PFOS. And yeah, so it still continues to follow the same trend for both birds A and both birds B. Relatively the same concentration for both batches. And so here's my summary. So both my quality control checks performed well, and they contained no contamination. Um, the negative control and the positive control both performed well. Um, I believe that the reason why we lost a lot of our compound was because of the molt patterns on the bird feathers. So the feathers naturally shed and regrow, which means that we probably weren't able to detect as much PFAS as we would for the blood and liver because, you know, they shed their feathers, and then they regrow them. So that is my conclusion. And so at the Lohmann laboratory, I was the first person to conduct research using feathers. And so I have a few recommendations for EPA. I recommend EPA to freeze dry all of their samples, and then instead of air drying like I did. And then I also recommend them to use NV-carb for their samples because that requires an extra step. And I couldn't conduct all that research in 8 weeks. So I didn't I didn't NV cart my samples. And so here I want to give a few acknowledgments to my mentors, Dr. Reiner Lohmann and Taylor Elpers. And I want to thank NOAA, Ocean Exploration Club, and the partnering universities for making this experience possible. And I want to thank the laboratory that I worked at for being very kind and very welcoming. And yeah. And so here's a summary of my time in Rhode Island. This is Kyla and I when we visited Providence, and and that we had so much fun. We also went to the aquarium, which was also really cool, and we visited multiple beaches in Rhode Island. And that's Taylor and I at the laboratory when I was extracting the bird feathers. All right, thank you. Thank you. So I like to open up the floor for any questions you guys might have.
You thought you were going to be rid of me, didn't you? Really interesting stuff, pretty cool. And I love that you learned so many benchtop techniques because that is a skill set that's definitely needed even in the field sense. And so that's going to make you really good at both sides of science, which I think is really unique. My question is, when you did the spike, if you found that you were losing quite a bit, what could you do or what would you do to correct your data? Or would you just have to throw out your entire analysis and start over?
So what I would possibly do is I wouldn't start over because I think all data is great data. So if my data was showing that I didn't have a higher PFAS concentration, I'll make sure I retrack my steps and look at exactly what I did for my methods to see if I, if I missed a step, because everything could really affect the data. So I would retrack on my steps. Did that answer your question?
Yes, thank you very much.
Thank you. All right.
Hey, Jayla.
Hey, Kyla.
Thanks for the feature. It was great. Well, being that we both worked at the University of Rhode Island this summer, I know that you had said that, being in the lab day in, day out, it was a lot. What would you say was like your just favorite moment or favorite time over the
Yeah,
summer?
that's a great
Well,
question.
in the lab, I guess.
So my best time was definitely hanging out with you and Willow. We had such an amazing time. We went on a boat and looked at like the type of how it operates. And then we also like explored Rhode Island on our own, and we visited beaches. Kyla and I like had like a beach lunch where during our lunch break, because our laboratory is very close to a beach. So we could literally, it's like a 5-minute walk. So for lunch one time, we walked down to the beach and just like sunbathed and just sat there and ate lunch and then went right back to work. So Yeah.
I was gonna ask, like, also, like, what was your favorite part of your project? What would you say was the biggest takeaway that you had?
My favorite part of my project was extracting the bird feathers because then I got to work directly with the animals, even though it was dead. So I got to work directly with the animals. And my biggest takeaway is like, so this is my first research project. I definitely realized how much effort and time and precision it takes to conduct research and how time consuming it could be. And I learned the importance of conducting research and how, because PFAS is a very big issue. PFAS is linked to cancer and also liver diseases. So it's something that affects all of us. And so it just brought awareness to PFAS because I didn't really know about PFAS before I started this internship. So I was able to conceptualize what is going on in the world and how I could improve it or help. So, thank you. Thank you. Thank you. Is there any more questions? Yes. I'm coming.
I mean, great presentation. So
Thank
this
you.
is your first research or intern?
Yes.
Well, one thing I like about what you put in your presentation was the evaluation, like mainly the like what you should have done better. I feel like that's a better way rather than saying like everything was perfect.
Mm-hmm.
It's better to show your like a better the second perspective
Exactly.
and see how you can improve because we're all, we make mistakes and we can be better. So yeah, and also I love this research because I'm always fascinated about the bioaccumulation and biomagnification of the sea life because nowadays it's like everything's polluted, the water's
Mm-hmm.
polluted, sea turtles eating plastics and it just accumulate into their bodies. So it's a really good research, I have to say.
Thank you
So
so
thank you.
much. Thank you so much. Yes. Thank you.
We do have a couple messages from online from your family, Jayla. Your mom said, "Can someone please tell Jayla her mom and aunt are watching and are very proud?" And you also have a proud cousin, and your dad said her dad is watching and proud of her too.